Yeah, so thanks, Pierre. Yes, and as Pierre just introduced me, I'm working at the National Institute of Aquatic Resources in Denmark. We work to, among other things, to integrate genetics and genomic information with resource management and sustainable fisheries and aquaculture production. And so what I'm present here is kind of the first views into some of our results on work we have done on the, and we are planning to do on the European flat oyster. And it's in a Nordic collaborative project. And so a lot of the things that I'm going to present is actually work that has been conducted by a colleague, Omer, who is doing a PhD on this project. So of course, what I'll do is to take all the credit for Omer's work and then refer all the tricky questions to Omer afterwards. Just to introduce to you our project and the framework. So this is a Nordic collaborative work. It involves partners from 6 different institutions in Denmark, Norway, and Sweden. And it's kind of geographically focused on this region that kind of unites the 3 countries on the entrance to the Baltic Sea. So we collaborate with colleagues from Gothenburg, the Swedish Environmental Research Institute, and University of Agder in southern Norway, and other colleagues of mine from DTU Agder. And then in addition to that, a lot of other people have collaborated and are still collaborating on this work, and we highly appreciate that. So this is also kind of an acknowledgement slide for this work. We have really enjoyed all the collaborative spirits in this oyster environment. So that's really nice. And also in particular, we have really benefited, and I think this NORA network is really a nice setting for integrating the work we do across countries. So also thank you to NORA and Pierre and Feline for organizing this. I think it's a very good opportunity to share information. And so a little bit of background to our project. We know, as Sylvie just also explained, of course, we know this patchy distribution of oyster beds, and they are also kind of patchily distributed in Denmark, Norway, and Sweden. We know that they have been here, they have been in the region for thousands of years. We have these classical kitchen midden excavations where deep layers, pretty thick layers of oyster shells have actually been found. And so they are thousands of years old. So they've been in the region for a very long time, most likely. But we also know that, for example, the Danish natural population that is currently here is a very young population. So it's only around 150 years old. And we know that because this particular fjord system where it's currently found, it was colonized. So it kind of has a history of being open and closed with connection to the marine environment. So the last kind of connection was established only 150 years ago. And so that meant that the area could be colonized. So that's a very young population. And in contrast, some of the coastlines around Sweden and Norway, they have been available for colonization for much longer, of course. Then many of these sites and populations are found in kind of unique environmental settings, as many other oyster beds, I'm sure, across the distribution range. So the Norwegian coastline is among the most northern edge of the distribution. We have the Swedish coastline, which is kind of sitting on the entrance towards the Baltic Sea. So this is in this transition zone between the North Sea and the Baltic Sea, which has been found in many species to be, it's a highly dynamic and transition zone. And we know that several species from many different taxa are genetically highly structured in this kind of environmental transition zone. The Danish population is found in a kind of semi-low to variable salinity conditions, and in particular, it's a very nutrient-rich area. So it receives the runoff from the farmland around the fjord system. We know relatively little about the kind of the relationship, genetic relationship, the evolutionary relationship between these different populations within the Nordic region. We know a little bit about their relationship to other European populations, but they have not been studied extensively. Now, Silvia just showed you some very nice slides that would complement this. These slides that I have here, extracted from the literature very well. So what we know from studies is that these kind of Scandinavian sites tend to be genetically different from other sites in Europe, and also that they seem to be more closely related, as Sylvia also just showed you to these kind of the Netherlands Wadden Sea system. So the Danish part of the Wadden Sea on the west coast of Denmark here used to have large oyster beds, but they are now completely gone, and it's only the Pacific oyster present there. But at least the genetic and evolutionary relationship, as Sylvia also explained, is kind of linked to this southern part of the Wadden Sea, North Sea system. But there's been relatively few studies, and no studies have really sampled extensively along this geographical region. What we also know is in the history of these populations, as in many other areas in Europe, that they have been subject to extensive translocation activities, at least in Denmark, and I think also for some of the Norwegian sites. In contrast, I think the general perception is that the Swedish coast has been less affected by these activities. So for Denmark, for example, we can see that they have received translocations inside this fjord system from many different areas, from the south and from the north. And of course, it's even more complex because these the sources for the translocations could have received translocations themselves in the history of these species. So of course, these are kind of human activities that would affect or could affect the genetic patterns we observe today in the natural populations. There is increased interest also in using, for this species and as in many other species, information about the kind of genetic diversity and connectivity, as Pierre also mentioned in the introduction, for kind of guiding like conservation and management initiatives. And so this is kind of a picture from the Danish fjord system, and these colors illustrate the densities of these oysters from relatively good monitoring programs established in this area. So we see, of course, that it's highly scattered geographically. So it's kind of these dense hotspots areas, and they tend to kind of be, of course, managed within an integrated system, also often with other management objectives such as Natura 2000 areas and so on. And of course, having some information about the distribution of genetic diversity, be it neutral or adaptive or whatever, and the connectivity between these areas would be informative for guiding these kind of management, more targeted management approaches. So the top map here is from the Norwegian coastline, and you also see similar types of very localized protected areas and no-take zones and so on. And so of course it's interesting for guiding of management to know a little bit more about how diversity is distributed within and between these areas. And then finally, we have interests in these kind of restoration and potential restocking programs, at least in Denmark and Sweden as well. So they are not currently ongoing, and that is partly also because of concerns over potential negative effects that these activities could have. So if you look at the history of how these Danish population has fluctuated over time, you see there are periods with very low abundance, and so in these periods, of course, it would be interesting to see whether you could supplement the population somehow. So this was a period where there was a complete ban on the fishing, and then fishing has been allowed back again. And the Danish system has had a long period where you were not allowed to translocate, mainly because the Danish fjord system was Bonamia free. That status has been lost not that long ago. So in principle now, this kind of Bonamia protection from translocation activities is actually no longer instated automatically in this system. So of course now we have the potential for kind of activities to help supplement the natural populations, but of course also concerns or at least awareness that we need more information about the potential effects that these activities could have in the systems. So our objectives here are kind of twofold. So we want to try and understand better the distribution of natural genetic diversity within this region, in particular in Scandinavia. And so, and also, as Sylvie also explained, to try and understand the demographic history of these populations, how and when they colonized the regions. And in particular, we're focusing on combining very dense geographical mapping of the population structure and the genetic diversity with using the high power we get from using genomic sequencing of the populations. That's one thing. That's kind of the basic scientific mapping of diversity and understanding of the evolution and ecology in the system. And then we're also aiming, because we're linking this very much to the management and we want to link it very much to the practical management and conservation also aspects of this work. So we also have another kind of leg in the project where we try to develop practical tools and some guidelines that can be used, guidelines coming out of our work and maybe also work of others along the lines of this kind of the lifetime of these projects. And so what we do is we do low coverage genome resequencing of these populations, combine it with dense, kind of focused dense mapping, sampling of the Scandinavian region, and then of course include some reference sites from outside to be able to interpret our local patterns. That's always important. And for that's one thing, the genome resequencing. And then we use kind of try to develop the microsatellite panels for parentage analysis in these hatchery settings. And that's been done before in other kind of context. Here, what we're trying to do here is we're linking it very much to the actual practices in the hatcheries to see how we can develop and kind of facilitate processes and procedures that could maximize genetic diversity from the procedures actually being used in the hatcheries. So this is our sampling program, or the sampling scheme we have currently, the samples we have collected now. So you see, of course, on the right-hand side, you have this dense sampling of the Scandinavian region, and then combining it with more scattered reference sites from other areas in Europe. And just to give you a little bit of insights into the type of data we're expecting to get from this. So we have just gotten the first batch of data out. So what we did was kind of a pilot study with just around 100 individuals. So few representatives from several of these putative populations. And then we did like a pilot sequencing just to see if our protocols basically worked and if we get the type of data out that we expect to get. So these results are very preliminary, and please treat them as such. But I'd still like to share it with you just to give you some idea about the patterns in the data. So we're aiming for, so these plots show you on the left-hand side, you have the sequencing depth. So how many sequencing reads do we actually have for one site in the genome? And the right-hand side tells you how much of the genome we have actually data for, how large a proportion of the genome we have data for each of the individuals. So one bar here is one individual. On the left-hand side, you see our target is about to get around 1x coverage for each individual. And you see that, well, there is some variability among these individuals, obviously. But we are pretty happy, I would say, overall, with this kind of output. There are some what we call outliers. So these are some individuals where we simply don't have, so we have less data than kind of expected. And I don't think this is, I mean, this is kind of normal in this type of data, that you get a lot of variability between individuals. And then, of course, some individuals will drop to quite low levels, and that has two effects. You see it here. One effect is that we have low coverage for individual sites, And we have also low proportion of genome covered. That you see that on the right-hand side. So we might reconsider these individuals, perhaps sequence a little bit more to get more data out of those particular individuals. But overall, we're quite happy and ready to scale up, I would say. A first look at the data coming out of this. So, and again, this is just a first pass analysis. So just to give you some idea. And of course, this also serves for us as kind of a check to see whether the data makes sense based on what we know from the literature. And so this is a PCA. One dot is then one oyster, and the different colors represent the different countries where they have been collected. And first thing, if we take the first axis in this PCA, we see these different groups. Adriatic group to the right, then a kind of Spanish, UK group, and then the bigger groups where we have included more individuals in the pilot sequencing from the Nordic, Scandinavian sites, and then the Netherlands sites. And if I can just block your view of these weird dots on the second axis in the PCA for a minute, I just say that these patterns on the first PCA correspond quite well to what we would expect from the literature, and also we just saw it in Sylvie's presentation, a split between Mediterranean and Atlantic in the south, and then we have the Scandinavian grouping with the Netherlands, and then the UK, some differences between the UK and Spain as well. So, but it's relatively few individuals from each site, so patterns are still not that clear. And I would say, of course, this is just a global view of the total diversity. So of course, more fine-scale patterns may be there if you analyze subsets of this data. And then of course, the weird ones on the second axis in the PCA, they are like from a hatchery-reared kind of population of oysters in Scotland. So they're not really grouping with the rest of the UK samples we have in here. And so I think this is something which is interesting and something to dig more into, of course, what's behind this, these patterns on a more genomic scale. That would be interesting to dig more into, and we will also include more individuals from this particular site in further analysis, of course. But I think it also shows you what could happen in kind of highly cultured environments to the diversity in this species and this population. And this is something we know, of course, from previous work as well, that it affects the diversity in the population. There's a lot more work to be done on this, also looking at potentially how related these individuals are and so on. And then another dot that just caught my attention when I just saw this plot a few weeks ago. Of course, there is one individual in here which has a Scandinavian genotype, because it groups with the Scandinavian and Netherlands cluster, but it has been sampled in the UK, southern parts of the coastline there. So first thing, of course, to ask is, did we mislabel this individual? And I cannot fully exclude that. We are still at that very early stage, so we need to rule out this possibility before we really dig into this. But also it could be indications of translocations, or maybe admixture from past translocations. And that is where we also need a little more information about the history of potential translocations between these different regions. And as Sylvie also explained, I mean, these groups are relatively distinct genetically, so we are able to really tease these things apart, this type of data. At least that's what we expect. And then, of course, what's next? We are going to dig more into this fine-scale patterns. We hope really that this genomic information, the genomic data will allow us to look at these patterns of structuring at even finer geographical scales than what we've been doing previously. Also because we have this dense geographical sampling program. So things like on the right-hand side, you have an example of what could be envisioned. That's the Swedish coastline, and that's kind of a look at the connectivity patterns among these different sites along the coastline. And this is what we could do if we have high statistical power with lots of genetic markers combined with dense geographical sampling. And so stuff like that would be interesting to pursue with this type of data once we start getting more data. And then so looking at also individual levels of variability, of course. And then, of course, also exploiting the full genomic information we get out to linking it to the genome that has been kindly shared by Anu with us as well. So getting this information about the genomic distribution of differentiation. And so we have these heterogeneous patterns comparing different populations in the region. And so don't pay too much attention to the actual values that you see in there. It's still first pass of data and so on. But what you should see is, of course, we have these heterogeneous patterns of differentiation. And something like this might be something that also showed up in Sylvie's analysis. I'm not sure it's the same part of the genome, but it could be a huge block which is highly differentiated. So, but more than that, and you see that you have, when you have full genomic information, of course, you can dig into the more file scale details as well. And that's what we're planning to do. So last bit, Pierre, can we please tell me if I'm running out of, when I'm running out of time?
Well, you ran out of time 2 minutes ago.
Okay, sorry about that. So I'll just spend a little bit of time here on the last part, which is the parentage analysis. We don't have that much data here to share. It's just to present to you the general ideas we have. So we're planning on optimizing these microsatellite panels for parentage analysis, and that is taking a little bit of effort because of various aspects you can see here. And then once we have it optimized, we are going to compare different practices in Danish and Swedish hatcheries to see what that affects, what kind of effects it has on the genetic diversity. So what actually happens in practice in the hatchery is that you have these potential parents in tanks, and then they spawn in different waves. And our idea is then to collect these kind of offspring here to analyze the parental contributions. How many parents do you actually get? Do you get more diversity by having many parents in one tank compared to fewer parents, and so on? Because we don't know. We know that it's probably highly heterogeneous contribution from different individuals, but we don't know this specific setting. And also what we want to do is compare across time, so sampling the larval pool, but then also at later stages, once they grow and once they settle, to see if there's kind of a transition happening within the hatchery. So in principle, you could have like non-random mortality in there, which could then even filter out the initial diversity to even lower levels at later stages once they get closer to being ready to being released, if that's what would be wanted. So early stage here as well for this work. So as I said, just please look at this preliminary data. We are very happy to reach out and collaborate with people who are interested, and also, as we just discussed before, with, for example, we still have capacities for integrating this with more data. If you could see your own kind of questions being relevant to address with this type of data, then please reach out to either Omia or me, and we'll try to see what we can do. So we're always looking for more work, of course. So thank you. Sorry, Pierre.
No problem. Thank you very much, Jakob.